the detailed general structure Protista, its types, and its classification. So let's start. So the kingdom Protista normally include the unicellular organism. So mostly the unicellular eukaryotes are included in the kingdom Protista. So what do you mean by unicellular eukaryotes? Means these are the eukaryotic cells having the nuclear membranes, having the membrane bond organelles. So as they are having the membrane bond organelles and nuclear membrane, they are called as the eukaryotic cells. And they are single cells. So normally they are made up of single cells. So as they are made up of single cell, these are called as unicellular eukaryotic cells and included in the term that is called as Protista. Now let's move the next point. Normally the mode of Protista, the classification of Protista is not based on the nutritional label. So it is mostly based on the 5 kingdom classification by Whitaker. It is based on 5 system classification. It is based on the 5 system classification given by Whitaker, but not on the nutritional point of view. This is the most important point, that Protista are not classified on nutritional value. These are classified on Whitaker's 5 system classification. Means we have Monera, then we have Protista, then we have Plantae, then we have fungi, then we have Animalia. So like this it has been classified, and it is all about of Whitaker classification. Let's discuss. So normally this classification is based on this particular group that is called as Whitaker mode of classifications. Now let's move to the next part. The term Protista was given by the scientist that is called as Ernest Haeckel. So Ernest Haeckel is that particular scientist who also discovered the 3 kingdom classification. The 3 kingdom classification also was given by the fellow that is called as Ernest Haeckel. Got it? So this is what we got, that the term Protista was coined by Ernest Haeckel. And these Protistans forms a connecting link. They normally forms a connecting link between Monera and multicellular organism. As we all know that Monera contains the unicellular prokaryotes and Protista contains the unicellular eukaryotes. So normally we have plants, normally we have fungi, and normally we have animals. So here what happens, these are unicellular prokaryotes, and these are multicellular prokaryotes. These are multicellular eukaryotes. So these are unicellular eukaryotes. So Protista forms a connecting link between unicellular and multicellular. So it is a connecting link, and also it forms a connecting link between prokaryotes and eukaryotes. Yes, and also it forms a connecting link between, that is called as unicellular and multicellular. So this Protista, for which this Protista sometimes called as the diversified kingdom. It is sometimes called as the diversified kingdom. Why it is called as the diversified kingdom? Because it has a diversified link with all type of kingdoms. It is linked with Monera, it is linked with Plantae, it is linked with fungi, it is also linked with Animalia. So for this it is called as the diversified kingdom. It is regarded as connecting link between prokaryotes and eukaryotes, connecting link between unicellular, multicellular, and also connecting link between, that is called as prokaryotes, eukaryotes. Got it? Clear? Now let's discuss. So yes, we know that these Protista present forms the connecting link between Plantae, between fungi, and also between Animalia. So they form a connecting link between Animalia, between Plantae, and also between fungi, as we have already discussed. Now let's discuss about this particular figure. So in this particular figure, what we got? We got that Monera is present as the unicellular organism. This is the Monera. So it is the unicellular prokaryotes. And these are the unicellular eukaryotes. Unicellular eukaryotes. And these all are the multicellular eukaryotes. Multicellular eukaryotes. Got it? So here Protista, they forms a connecting link between Monera and all this group. So Protista forms a connecting link between unicellular prokaryotes, two unicellular, that is multicellular eukaryotes, it all forms a connecting link between unicellular and multicellular. So normally we find this protista, they form a connecting link between unicellular and this is the multicellular. Got it? And so protista forms the connecting link not only between these two, it also forms connecting link between this and also connecting link between this. Got it clear? So this is how protista is regarded as the most diversified kingdom. Now let's discuss its general character. So we have discussed that protista are the ancestor of all eukaryotes. Yes, we have discussed. And multicellular eukaryotes includes plant type, fungi and animalia. Also we discussed. And let's discuss what protista do include. In this kingdom, which group of organisms are included. So mostly photosynthetic protista means autotrophic protista. So these are the autotrophic protista, means they have photosynthetic pigments to synthesize the food. Example like dinoflagellates, examples like diatoms or desmides, and examples are euglenoids because they all contains pigments. They all contains pigments, and due to the pigment, they helps in photosynthesis. They undergo photosynthesis. We'll discuss later details about this, for which they are regarded as autotrophic or photosynthetic organism. Now let's move the second group, that is called as the consumer group or decomposer. These are saprophytic. These are saprophytic. These are saprophytic means they depends on dead organic matter. That depends on dead organic matter. So these are saprophytic that depends on dead organic matter. The best example is cellular slime molds or as well as slime molds. We'll discuss later. This is a type of slime molds. This is slime molds present. The third type is called as animal protista. This is called as animal protista. The third type is called as animal protista. This animal protista are called as protozoans. The best example is Euglenoids, means they are having flagella. Then we have Sarcodina, means they are having pseudopodia as the locomotory organelles. Then Sporozoa, they don't have any locomotory organ, they produce spores, and these spores liberate and get dispersed out. And Ciliated Protista means they have cilia as the locomotory organs. So these four groups of animal protista included under this kingdom, that is called as protista. Now discuss about the general character. So normally all the protista unicellular eukaryotic organisms as we have discussed, and some are colonial without cellular differentiation. So what does this mean? So some group are found in colonies. They are found in colonies like Plasmodium. So they are found in colonies. So some groups are found in colonies. They are colonial. And some groups are found in unicellular. Most of the groups are unicellular. So these are unicellular. So most of the groups are unicellular, but some are found colonial. But some are found as colonial. Why? Now there is not much cellular differentiation, means no tissue system is present. No tissue system, that means no tissue system. So in this particular group, we don't find tissue system present because without cellular differentiation means they don't have any tissue system in this particular group of protista. Clear? Now let's move. Yes, they don't have the tissue level of is absent totally, organization of tissue level is totally absent. And they're mostly aquatic, means they're water loving. They are found in waters. So most of them are found in water. Either they are found in fresh water or they are found in marine water. So for which they are aquatic in the mode of habitat. Now the cell structure of eukaryotic types, because as we all know that all the eukaryotic cells, they have a cell membrane, they have a nuclear membrane, and they have membrane bond organelles. So these membrane bond organelles with cell membrane and nuclear membrane are regarded as the eukaryotic cells. So this is the eukaryotic cells. And they find the nucleus present. This is the nucleus present. And these are membrane bond organelles are present. These are the organelles are present. Also we find here in the cytoplasm, we find ATS ribosomes also present. ATS ribosome is present. Inside mitochondria and chloroplast, we find 70S ribosomes also present. So all these characters rectify them as the eukaryotic character, but they are unicellular, single cell organism because they don't have tissue organization. So the body is completely made up of single cell. So as it is made of single cell, so mostly they are cellular mode, protoplasmic mode of body organization is found to be present. Got it? So this is that they are eukaryotic in nature. Now let's move the next part, that is this group of organism contains 80S ribosome in the cytoplasm, and also they contain 70S ribosomes. And the very important fact is that they have a cell wall, and the cell wall present most of the cell wall present, the cell wall is cellulosic. They have a cell wall, and the cell wall is made up of cellulose. The cell wall is made up of cellulose. Got it? So it is the plant-like nature. So it is the advanced form, this Protista. Now, the flagella and cilia are present as the locomotory organs. Means in cilia, in 5 we find flagella either present or we find the appendages present, that is called as cilia. So the flagella present or we find the cilia are present. And these are the locomotory organs. Locomotory organelles. Got it? And the mode of pattern of arrangement is 9+2. So what does this 9+2 indicates? Means suppose we'll take the anatomy of any cilia, or that is called as flagella. What we find that in the periphery we find this doublet 9 microtubules are present. 9 group of microtubules, 1, 2, 3, 4, 5, 6, 7, 8, 9. And these are called as the peripheral doublet microtubules. And at the center we find 2 individual singlet microtubules. These are called as singlet. So the doublet and singlet is 2 and doublet is 9 in number. And these are made up of microtubules. These all are made up of microtubules. And these microtubules are made up of tubulin proteins. They are mostly made up of a cell protein that is called as tubulin protein. So this type of arrangement is called as 9+2 arrangement, and it is the pattern of eukaryotic cells. So it is the basic character of a eukaryotic cells. So the cilia, either cilia or flagella, they both contains 9+2 arrangement of singlet and doublet microtubules present on it. Now let's move into the next part. Its next character, that is, we find the microtubules are made up of tubulin proteins, and the movement is either by means of cilia, flagella, or by means of false foot. If the movement is by means of this false foot-like structure present, and these are called as pseudopodia. And mostly pseudopodia are found to be present in Sarcodina group, that is amoeba. So this is the most vital part of locomotion. On the basis of locomotion, the animal Protista is being classified. Now we find that the fastest movement is the ciliary mode of movement. So the movement by cilia mode is regarded as the fastest movement. Yes, good. And next we find the mode of nutrition is either photosynthetic, means these are by having plant nature. Why they plant nature? Because they do have pigments. So as they do have pigment, they have plant nature. Or either they are by animal nature, ingestion, means they ingest the food materials, they engulf the food materials. So the food material is engulfed and being dissolved within the cell. And either they may be having, that is called as saproboicoparasitic, means on the surface of the organism or dead organic matter, they secrete enzymes, and these enzymes dissolves the food materials, and these food materials are absorbed by the organism. And so mode of absorption, the nutrition is by absorptive. So absorption of nutrition or absorption of food materials, they helps in the food materials preparation, nutrition. Now in some we find both type of nutrition present, that is photosynthetic and saprobiotic, that is mostly found in Euglena. Euglena we find both type of nutrition, that is plant type and animal type, that is photosynthetic, and also we find saprophytic. Got it? Now move on the reproduction, either reproduction asexual mode without the use of gametes, or by means of sexual modes with the use of gametes. And let's move into its life cycles. The life cycle is classified into 2 groups, either zygotic meiosis or gametic meiosis. This is a very important part. Let's discuss about zygotic meiosis. Suppose an organism is haploid. For example, organism is haploid. So suppose the organism, the plant body of the organism is haploid. So this organism by means of mitosis produce gametes. So these are the gametes which are also N and N. These are the gametes produced. And these gametes undergo fertilization. Yes, these gametes undergo fertilization. After fertilization, they produce the zygote. After fertilization, they produce the zygote, which is 2N. The condition is that the plant body, the parent is haploid, but the zygote so produced is a diploid. So to form the plant, the parent plant, they undergo meiosis. So meiosis takes place on zygote. So meiosis takes place on zygote. So zygotic meiosis. So zygotic meiosis is mostly present in haploid set of organisms. So this type of meiosis is found to be present in this particular group, that is Protista. We'll discuss later, that is diatoms, and in desmites or dinoflagellates we find all this type of reproduction is found to be present. Now let's talk about gametic meiosis. In gametic meiosis, if the plant body is diploid, so they produce gametes. They produce gametes by meiosis. So this type is called as gametic meiosis. So if the plant body is diploid, they go for gametic meiosis, and if the plant body is haploid, they go for zygotic meiosis. So this is about the life cycle of this particular group, that is called as Protista. Now let's discuss about other factors. They are decomposer, yes, saprophytic. They are photosynthetic. They are parasitic, means being parasitic, they are pathogenic, disease causing. Yes, they are disease causing organism, because parasitic are what? Pathogenic. Parasitic are mostly what? Pathogenic in nature. So as they are disease causing, they cause a dysentery disease, malaria disease, sleeping sickness. These are caused by the animal protozoans. Now let's discuss about some practice questions. So we have completed the basic general character of the kingdom, that is called as Protista. Later we'll discuss about its particular kingdom, subkingdoms, is Photosynthetic Protista, or that is called as consumer Protista, or animal Protista. We'll discuss later. First we'll complete the general character, then we'll go for that. Let's discuss the practice questions. Suppose the assessment is saying that Protista forms a diversified kingdom. Yes, because they have all these characters. Yes. Why they form? Due to these reasons. So when we go to the options, the best option is this. Yes, both A and B are true, and R is the correct explanation to that of A. Next questions. Classification of Protista is difficult. Yes, it is very difficult because the members have all these features, for which photosynthetic algae, saprophytic slime molds, heterotrophic protozoans are included. So the A is right and is the correct explanation. Now the main reason for difficult in classifying provide, it is very difficult to classify because they are both plant character and animal character, so we are unable to differentiate. They all reproduce only asexually or complete auto. No, these are not the points. So the best example, the answer is, that is, they exhibit both plant-like structure and animal-like traits. The most diversified kingdom, as we all know, Protista. Yes. The kinetic link between unicellular multicellular, as we discussed, that is called as Protista. Yes. The kinetic link between prokaryotes and eukaryotes, yes, we have discussed, that is called as Protista. So if you want to practice more questions on NEET level or any entrance point of view, you can go to this particular link and practice questions, or you can go to this particular link and give direct online questions. You can also go for doubt clearing classes to this particular site, or if you want to download the mind maps, worksheets, MCQs, PDF, you can go to this particular link. Here what happen, you will get per chapter you will get 1000 plus questions. So it's an ample amount of questions present, easier for practice. And also with the questions you will find the explanations are present. So easy explanations are present, you can go for it. If you want to correct your doubt by direct through online, or by direct you can contact with this number, or if you want any PDF, direct PDF and question materials, you can go to this particular channels. Okay, hope you all enjoyed. Thank you. Today we are going to study about photosynthetic protista. So these are the protista where we find that they are plant-like structure, means they have plant-like characters. They are the plant-like characters protista, means they have pigments. They have pigments so that they can produce their own food, and also they have the cell wall. And here we find the cell wall which is cellulosic, So as we are going to discuss the details about the photosynthetic protista, so let's start one by one. So we know that they are popularly called as the protistian algae. So sometimes we do call them as protistian algae. This is called as the protistian algae. And most of them are phytoplankton. So what do you mean by phytoplanktons? Means they float on the surface of water. Normally they float on the surface of water. So these are the plant that floats on the surface of water. So those organisms that floats on the surface of water, they are called as phytoplanktons. It is plants, so they are called as phytoplankton. If it is animals, they are called as zooplanktons. And let's discuss one by one. The first example we are going to take, that is called as diatoms. Also included in it is called as desmites. Let's start. Now the first photosynthetic protista we are going to discuss, that is called as diatoms. So what are diatoms? Diatoms are normally called as golden brown photosynthetic protista. Very important. These are called as golden brown. The body color is golden brown, and they are photosynthetic protista, means they can prepare their own food, and they are included under, that is called as chrysophytes. So they are included under chrysophytes, and also these chrysophytes includes both diatoms and desmites. So chrysophytes, under chrysophytes we find diatoms is present, also desmite, but we are going to discuss only diatoms. Let's move. These diatoms are both aquatic, found in water, and terrestrial, found in land. So they are found on land and also they are found in water. And the water may be of two types. Either they are freshwater, or some are of marine water, but most of them are marine water. Got it? Now, yes, some are marine, but mostly they are called as marine water. They are marine in nature because they love much as marine life. They support marine life conditions, so most of them are marine in nature. And normally they are very microscopic, they are very minute, and they have different colors. Varying colors are present on it. Due to the presence of pigments, they have varying colors, different colors, and they are microscopic organisms. They are very minute organisms, and mostly they are unicellular. What unicellular? The unicellular eukaryotes. These are unicellular eukaryotic organisms. So normally these are unicellular eukaryotic organisms. They have many colors and they are very small, very minute, can be seen only under the microscope, means they are very microscopic, very small in nature. These diatoms may have pseudofilaments and colonies. Normally they don't have colonies, but these diatoms sometimes have this group of filaments. So these filaments are actually not true filaments. They only join, but at other time, at some time they will isolate into unicellular form. So this type is either called as their colonial or pseudofilaments, means false filament. It is not a true filament-like structure. And we move that these diatoms, they don't have flagella. Yes, they don't have flagella. Only the flagella is found only in the reproductive stage. So the vegetative state lack flagella. So vegetative cell lacks flagella. But reproductives do have flagella. And they may be free floating. Yes, we have discussed they are phytoplanktons, means they are mostly found on the surface of water, floating on the surface of water. So those organisms that floats on the surface of water are called as phytoplanktons. So the iron always floats on the surface of water, for which we do call them as the phytoplanktons. These are called as the phytoplanktons. Due to the presence of lightweight lipid, why they float on water? Because the cell wall and the cell membrane, they have lipids. So the boundary lipids, due to the presence of lightweight lipid, they are able to float. So they only float due to the presence of lightweight lipids present on their boundary or on the cell membranes. And the cell wall is covered with silica and transparent silica. That silica very transparent, called as transparent siliceous cells. Means suppose this is the diatom present. So externally the diatom is covered with silica. Externally the diatoms is covered with silica. And this silica is very transparent in nature. So normally diatoms, they have the cell wall. Yes, they have cell wall. Above cell wall they have a covering called silicated covering. And this silica is very transparent in nature. So sometimes we do call them as transparent siliceous cell. And this transparent siliceous cell is sometimes known as frustules. Yes, the transparent siliceous cell which is present outside these plates are sometimes called as frustules because these are made up of silica and they are very transparent. And these diatoms covering silica is given a special name, that is called as frustules. Depending upon the symmetry, means division of body layer, diatoms may be pennate type or centric type. So what is pennate type? If the body is bilateral symmetry, if body is divided only in two forms, it is called as bilateral symmetry, it is called as pennate type. So pennate type means it is bilateral symmetry. So body is bilateral symmetry. So when the body is bilateral symmetry, they are called as pennate type. And normally in this pennate type, The body is bilateral symmetry as we have discussed now, and the best example is called as navicula. So navicula is that diatoms where it is called as the pennant type. The other name of pennant type is bilateral symmetry, means the body is divided into two equal halves. So it is called as bilateral symmetry, and is mostly seen in the example that is called as navicular diatoms. And in some, the body is centric. The body is centric. In some, we find the body centric. If the body is centric, it is called as radial symmetry. This is radial symmetry. Got it? If it is pennant type, it is called as bilateral symmetry. It is centric type, so it is called as radial symmetry. So this radial symmetry is mostly found to be present in the centric organism, a centric form. And the best example is called as Melocyra. So Melocyra is the best example. Navicula is for pennant type and Melocyra is for, that is, centric type. Let's move. The cell wall is differentiated into two halves. Normally, when we discuss about the cell wall, we find that the cell wall is differentiated into two layers. Suppose this is the cell wall is present. Another layer present, which is present just below it. Just below it. Just below it. So the cell wall is covered with silica, and these silica are normally present in this format. This format. Got it? And inside we find the cell membrane present. Inside we find the cell membrane present, and within the cell membrane we find the nucleus are present. The nucleus present, and around the nucleus we find the membrane bound organelles are present. We'll discuss later. Vacuoles present, everything. So normally this is the condition of your, that is called as the diatom cell wall. So the upper part of the cell wall is called as epitheca. Epitheca. So theca is the covering of the cell wall, and the lower part is called as hypotheca. Hypotheca. Got it? And they look like a soap box like structure. This is a very important question. They look like a soap box like structure, and both this wall is together is called as theca. So both the wall is called as theca. So as we all know that the diatoms, we find a modified cell wall present, where we find this covering of cell wall with impregnated silica is called as theca, and this theca is of two types. One is upper covering, that is called as epitheca, which covers the lower one, that is called as hypotheca, which looks like a soap box arrangement, and within that we find the cell membrane present. Inside we find the cell membrane present, and we find the cytoplasm present. Yes, here we find the cytoplasms are present, and also within it the cell organelles are present. So this is the basic structure. There is a modified structure of this particular diatoms. So normally what happens, the outer wall is called as epitheca, as we have discussed, and the lower wall is called as hypotheca, as we discussed. So theca is the covering. Theca is the impregnated silica covering present on the cell wall. Upper one is called as epi, and lower one is called as hypo. And the arrangement looks like a soap box arrangement. So diatom's best example is that soap box. So if the question comes, soap box arrangement is found in which group of organism, that is the best example, that is called as diatom. So this is, here is the example, and it looks like soap box like arrangement. As we find here what? Now normally here we saw the structure that this is the centric type, this is the soap box type, this is epitheca. The upper one is called as epitheca. The lower one is called as hypotheca. Yes, hypotheca. And they are joined, and this is the intercalary bands are present outsidely. And here also we find the centric type. This is centric type, the central area present. So this is the example of this particular group, that is called as diatoms. So these are this is centric type. This is bilateral, that is a soap type. So these are different form of diatoms. Okay, the cell wall encloses a peripheral layer of cytoplasm. What does this mean? For example, as we have discussed that the cell wall is made up of this epitheca and hypotheca. Yes, epitheca. So within the cell wall we find the cell membrane present. So this is the cell membrane present. So inside we find the cell membrane is present, and these are the cell wall. The cell wall in the form of epitheca and the cell wall in the form of hypotheca. Got it? Hypotheca. So within it we find a vacuole is present at the center. So at the center we find vacuoles are present. So as the vacuole takes the major part at the center, so what happens? The cytoplasm with its organelles moves to the periphery. The cytoplasm moves to the periphery. Got it? the cytoplasm moves to the peripheral layer. So when the cytoplasm moves to the peripheral layer, this condition is called as primordial utricles. Primordial utricles. Got it? Why? Due to the centermost part of vacuoles present, so all the cytoplasm moves towards the periphery, and this type of arrangement is called as primordial utricles. So it is the peripheral arrangement of cytoplasm near to the cell wall. So this arrangement is mostly present in this particular group, that is called as the diatoms. Got it? So we have discussed. Okay. Now let's see. So this peripheral layer is called as primordial utricles, as we have told. And it surrounds the last central vacuoles. Yes, we have discussed that it surrounds the middle where last central vacuole is present. So the cytoplasm moves to the periphery. Due to the movement of cytoplasm to the periphery, what happened? Now this condition is called as primordial utricle due to the last central vacuoles. So now the question comes, sir, if the central vacuole is present at the center, then where do the nucleus will present? Yes, the nucleus also hangs upon and on the vacuoles. Got it? The nucleus hangs upon the vacuole. We'll discuss later. Got it? Now let's move. The nucleus lies in the central part of the vacuoles. Let's see how the nucleus is present on this particular vacuoles. So as we have discussed that this is the outermost layer, that is called as the epitheca, and this is the lowermost layer, that is called as the hypotheca. So we got two most layers, epitheca and hypotheca. And for example, this is the cell membrane present as usual. Now the condition comes that at the center we find the vacuoles present. Got it? We find the vacuoles present. So when the vacuole is present at the center, so normally what happens? Now all the cytoplasm moves to the periphery with its organelle. So most of the cytoplasm with its organelle moves to the periphery. So when the cytoplasm moves to the periphery, so this condition is called as primordial utricles. So where do the nucleus will arrange? Yes. So normally what happens? The nucleus arrange at the center. The nucleus is arranged at the center. So this is the nucleus. So nucleus is present on vacuoles, and these are attached to the vacuoles wall with the help of small thread-like structure. So these thread-like structures called as cytoplasmic strand. Means how they arrange. For example, let's take, for example, this is the vacuole present. Got it? Suppose this is the vacuole present, and for example, this is the nucleus present. Got it? So this is the nucleus present. So nucleus is present on the surface of the vacuoles. So it is joined with the vacuolar membrane by cytoplasmic strands. And these cytoplasmic strands, these are what? These are called as cytoplasmic strand. Cytoplasmic strand. Got it? So this cytoplasmic strand hangs the nucleus on the surface of the vacuole, and this is attached with the membrane of the vacuole, that is called as the tonoplast. We know the membrane of the vacuole is called as tonoplast. So from the tonoplast we find cytoplasmic strings are present that holds the nucleus, which hangs. So sometimes it is called as hanging nucleus. So it is sometimes called as the hanging nucleus. So nucleus hangs on the surface of the vacuoles, and it is attached on its periphery with the help of cytoplasmic strand, which are attached to that of tonoplast. So this is the arrangement of nucleus, which lies at the central vacuoles. It lies on the surface of the central vacuoles. Got it clear? Now let's see. Next point, that it is suspended with the help of, yes, it is suspended with the help of cytoplasmic strands. So these cytoplasmic strands are present which holds the nucleus, and these cytoplasmic strands comes from the vacuolar regions. Got it? So vacuole gives out the cytoplasmic strand that holds the nucleus at the center. So vacuole is present at the center, nucleus is also present at the center, but nucleus hangs on the surface of the vacuoles. Got it? Now let's move. Mode of nutrition is photosynthetic, or it is holophytic. So phototropic mode of nutrition is one because they do contain pigments. Let's discuss. The photosynthetic pigment is present is chlorophyll A. Yes, due to this special pigment, they can prepare their own food. They also contains chlorophyll C. Very important questions. They contains chlorophyll A. They also contains chlorophyll C also. And In addition to chlorophyll A and C, they also contains beta carotene and fucoxanthin. The fucoxanthin provide body color. It is golden brown. So this golden brown is due to the presence of this particular fucoxanthin that provides body color. And beta carotene is also found to be present. So we have to remember chlorophyll A, chlorophyll C, beta carotene and fucoxanthin is present, which helps in photosynthesis and also helps in imparting the body color. Got it? So these are the pigments present. Now, they contain the special pigments that is called as diatoxanthin. Very important. They contain two special pigments. One is called diatoxanthins. Another one is called diadenoxanthins. Another one is called as diadenoxanthins. These are the two special pigments present. The two carotenoid pigments present special pigments which are the derivatives of this fucoxanthin, that is called as diatoxanthin. Another one is called as diadenoxanthin. Got it? So these are the special pigments found in diatoms. The reserve food materials is oil, yes, and also starch. And starch is present in the form of polysaccharide called as leucocene or chrysolaminarin. Very important. The reserve food material is oil, yes. In addition to oil, they also contains polysaccharides, and the polysaccharide present in diatoms is called as leucocene or is called as chrysolaminarin. So we should remember this particular point. It is very important in NEET exam point of view. And sometimes they contain phosphate granules or volutin granules are present in addition to, but these volutin granules are present in trace amount. They are responsible. The very important point is that 50% of photosynthesis or organic matter in the world, in the biosphere, is given by this particular diatoms. Very important questions. So diatoms gives 50% of organic matter of photosynthesis to our biosphere. So this particular diatom, due to the presence of pigments and due to its maximum speciality and due to its habitat in the marine form, they gives out maximum oxygen and also they give out maximum organic materials to the biosphere. And the movement is by mucilous propulsions, means the body moves. Suppose it's the body, so they have certain mucus present, and through this mucus they move slowly with the help of mucus contraction, body contraction relaxation, they move on the mucus, and this movement of mucus is called as mucilous propulsions. The movement is by mucilous propulsion. Very important. Now, they are mostly asexual mode of reproduction. They prefer mostly asexual mode of reproduction. That is mostly they use, that is called as binary fission. So binary fission is its normal mode of reproduction. It is the asexual mode of reproductions. So the mode of reproduction is mostly binary fission, and in binary fission what happens, one half of the cell is retained by each of the daughter cells so formed. So let's see what happens. Let's see what happens. Now normally, suppose this is the diatoms present. Got it? Suppose this is the diatoms present. So this is the diatoms present. Got it? And for example, take it that this is the parent diameter. This is the parent diatoms present. Got it? This is the parent. So when binary fission takes place, what happens? One half of the parent epitheca is retained in the daughter. So one half of the parent epitheca is retained on the daughter. Similarly, the other half is also retained in the daughter. That is, hypotheca is also retained in the daughter. So these are the parental part. Now what happens? This is the epitheca part. This is the epitheca part is retained in one daughter, and this is the hypotheca part. And the hypotheca part which is retained in another part. Now what happens? A new format is formed. A new cell wall daughter is arranged. If it is epitheca is the parent, then the new format will be hypotheca. So this is the daughter. This is the new. This is the new. Similarly, here also the new. So parent cell wall is retained and only the daughter cell wall is being formed, and this is the new. So this is how what happens. Now the cell wall, the reproduction do takes place like this. Got it? So these are the daughter. So each daughter retains some half of the parent cell wall. One daughter cells retain the epitheca of the parent and produce a new hypotheca, and other part retains the hypotheca parts and produce the new epitheca part. This is how the binary mode of reproduction do takes place. Got it? Now let's move. So binary one half of the cell wall is retained and the other half is also daughter is so formed. Now The other half of the cell was secreted new. Yes, we have discussed now. It is secreted new. Sometimes in adverse condition, they produce a cyst-like spore that is called a statospore. So in adverse condition, to withstand the adverse condition, they produce a resting spore that is called a statospore. Very important. Statospores are found mostly in centric diatoms. So we have discussed Navicula and Melocyra. Melocyra is centric. So in centric diatoms, mostly statospores are found to be present. Very important questions. Now they also reproduce sexually. They also can reproduce sexually. But sexual reproduction is isogamous to anisogamous. So what is isogamous and what is anisogamous? If gamete means union, what does gamete means? Union. If the gametes are same in their size and union, it is called as isogamy. Isogamy. Got it? If the gametes are irregular in their size of union, these are called as anisogamy. Anisogamy. Got it? If one of the gamete is non-motile and other gamete is motile, suppose one gamete is motile, another gamete is non-motile, another one is non-motile. If this is the condition, then it is called as oogamy. So this is called as oogamy. So three mode of sexual reproduction is found to be present. Yes, they do produce gametes. The gametes are of same size. Either the gametes are of different size. Either the gametes are of different size, but one is motile, another one is immotile. So this particular type is called as oogamy. Irregular gamete size is called as anisogamy, and regular gamete size is called as isogamy. So this is the sexual mode of reproduction. So sexual reproduction, yes, they involve gametic meiosis. Yes, they involve gametic meiosis. Why? Because the plant body we have discussed, it is 2n. If it is 2n, it will produce gamete. It will produce gamete. To produce gamete, they undergoes meiosis. So now we came to know that diatoms are diploid. So as they are diploid, so they go for that is called as gametic meiosis, as we have already discussed. Now let's move. Diatoms generally are diploid. Yes, due to the diploid, any diploid organism, they will go for gametic meiosis. As we all are, animals are diploid. The diploid animals go for gametic meiosis. But if it haploid, then they go for zygotic meiosis. So diplontic life cycle is found to be present in its diatoms. So diplontic life cycle means most part of the life cycle is of 2n type, and some part is less part is of n type. So if the maximum part is 2n type, it is called as diplontic. If the maximum part will haploid type, it will call as haplontic. But in diatoms, it is diploid. For it is called as diplontic mode of life cycles. Now move on to special facts present in diatoms, like the silica cells of dead diatoms are nearly indestructible, unable to destroy. Yes. For example, let's take an example. Suppose this is the water present. The water body is present. And we know diatoms are floating on the surface of water. Diatoms are floating on the surface of water. And this is the bottom of the ocean, that is called as benthic. Suppose when the diatoms die, they fall down. They move down. And there what happens, the silica gets deposited. When the silica gets deposited, they form mountains. And these mountains are mountains of silica. These are mountains of silica. These are mountains of silica. And these silica mountains are called as dolomite. Sometimes we do call them as dolomite. And this dolomite is very gritty, is very rough, very gritty, very hard. So as they are very gritty, very hard, and other characters are also present for which it is mostly used in our economic importance. Let's discuss. So we know when the diatoms gets, when the dead, the accumulates at the sea bed, a benthic region, and huge rocks are deposited. Hard cells of diatoms are deposited to form mountains, and these are called as diatomaceous earth. These mountains so formed inside the earth are called as diatomaceous earth. Now what happened? So this diatomaceous earth is mined as a whitish powder. So when we, for economic importance, when we extract the diatoms, these are white powder-like structure. So we extract in the form of white powder-like structure from the bottom of the sea for economic importance. This white powder is called as diatomite or diatomaceous earth or Kessel-Gur. Got it? The white powder so extracted from the mountains or from the dolomite or from the diatomaceous earth are sometimes called as Kesalgur or sometimes called as diatomaceous earth or sometimes called as diatomite. This white powder, very important economic importance powder. Let's discuss what is this white powder come. They're very rough and they're very gritty, and also it is used in filters in brewing industries. In brewing industry, in alcoholic fermentation industry, we find these are acts as filters. So we use in industries, brewing industries. It is also used in sugar industries for sugar refineries to filter sugars. It is also used to polish metals, toothpaste, and bricks. And these are also used as insulating bricks because these dolomites or these diatoms of earth are soundproof. So as they are soundproof, they are used in insulating bricks, mostly used in film halls or cinema halls. Also, we find these do help in manufacture dynamite. Sometimes, not most of them, sometimes some of them are used in manufacture dynamites. It is used as for water glass, sodium silicate, and strong acid formations. So many economic importance of this dolomite or diatomaceous earth is found. That is, they have helps in formation of water glass. They are used in the sodium silicate formations. They are used to produce strong acids. So these are the different economic importance. Also, sometimes they are used in paints to increase the visibility. Yes, they're very good indicators of water pollutions. Yes, if water is polluted, then we don't find diatoms are present. So due to the absence of diatoms, we can indicate that the water is polluted. So they acts as a good indicator of water pollution. Very important part. And also, they have oxospores are zygote formed during sexual event in some diatomaceous. In some diatoms, we find a sexual spore is produced. Normally, spores are produced by asexual reproduction, but in some diatoms, sexual spores are produced, and these sexual spores are called as oxospores. So we do call them as what? Now oxospores, and these are called as zygote formation of spores. And this oxospores helps in restoration of size. It helps in formation or resizing. It helps in restoring its original size. So oxospores helps in restoration, and oxospore is a key spore. It is called as it is a sexual spore. So oxospores are called as sexual spore. Common examples of diatoms, that is called Triceratium, Melocyra, as we have discussed, Navicula and Cymbala. Navicula and Cymbala. So these are the common examples of diatoms. So we have discussed the major facts, the general character of diatoms. So now let's discuss some question practice. Let's go for certain question practice. Now let's discuss about certain practice questions on diatoms. The first question is assertion reasoning. That diatoms are regarded as the pearls of the ocean. Yes, because of the siliceous walls. Yes, this is right. The cell walls are made up of cellulose, pectin, which dissolves. So this statement is not right. So the correct answer A is true, but R is false. Now move to the next questions. Diet was important contribution of marine. Yes, that's right. The fluctuations composed of silica resist rapidly decompose, and when diatoms die, they remain settled to the bottom of silica deposit. Yes, both are right. For which it is true. Got it. So now move on to the next. Diatoms are among the most important primary producers in aquatic ecosystem because of the photosynthetic efficiency and siliceous cells. Yes, it's true. After that, the silica fructose accumulates. Yes, they both are true. Diatoms are parasitic unicellular organisms. The cell wall is made of cellulose and pectin. The cells forms deposits known as dolomite. Yes, this is the right answer. Got it. But the fourth question, soap box arrangement is found diet question diatoms. Yes. And that is diatomite is found in diatoms. Yes. Okay. So there are a lot of questions present. So you can go to this particular site. In this particular site, you will get ample amount of questions, about 1000 question plus. If you want to go for diet online exam, you can know your score, know your rank. And if you want any doubt clearing classes, you can go for online classes, this particular link. If you want to directly download the PDF materials, the mind maps, the worksheets or the MCQs, you can go to this particular link. And mostly per chapter we are having 500 to 1000 questions you get per chapter. You can contact directly for any help, or you can directly download from our channels. So hope you all enjoyed about diatoms. Thank you. So let's discuss about dinoflagellates. Last class we have discussed about diatoms, one type of photosynthetic protista. Today also we are going to discuss dinoflagellate, which is one type of photosynthetic protista, but it is golden brown in color. The body color is golden brown. So as it is golden brown in nature, so it is sometimes called as golden brown photosynthetic protista. Why it is golden brown? Because due to the presence of pigment, different type of pigments. And these are included under the class that is called as Dinophyceae and under Pyrophyta. Got it? They belongs to the class that is called as Dinophyceae and is included under that is called as Pyrophyta. They are mostly marine in nature. Yes, they are mostly marine. Very few are freshwater. Very few are freshwater, but most of them are marine in nature. They appear in different colors. The body color is of different colors because they have variation in pigments. They are red color, they may be yellow color, they may be green color, they may be brown color, they may be blue color. Yes, due to variation of pigments, they have different variation of color, body coloration. And mostly these pigments do help in body coloration. So the body normally the cell presents pigment to control this body coloration. Now let's move. These dinoflagellates are mostly unicellular and they have contains flagella. Got it? So these are called as unicellular flagellated diatoms. Why it called as dinoflagellates? Because they have two flagella. Flagella 1, flagella 2. So these are the golden brown photosynthetic protista which are unicellular eukaryotes. These are unicellular and also these are eukaryotes. These are unicellular eukaryotes having one is called as the two type of flagella is found to be present. And normally in some the mode of locomotion is either non-motile. So when it is non-motile, the mode of locomotion will be amoeboid type or they move in groups that is called as palmelloid type. So mostly the locomotion is motile due to flagella. Mostly they are motile. But in some groups, different mode of locomotion is present like amoeboid type or palmelloid type. And they are mostly, as we have discussed, they are mostly marine, but very few are freshwater in nature. Let's move. They have a rigid coat called theca. For example, suppose this is the diatoms present, dinoflagellates present. So these are dinoflagellates body present. Got it? So around the body, they contains a thick covering. And these coverings present around the body are plates. They are of plates. Plates of what? Now, plates of cellulose and pectin. So these plates of cellulose and pectins are otherwise called as theca. So the dinoflagellate's body is covered with a theca which are made up of plates and these plates are made up of cellulose and pectin. Now let's move. The theca consists of too many articulated or sculptured plates of cellulose and pectin. Yes, as we have discussed, suppose this is the body present. Suppose the body present. So these plates are present one above the other as sculptured, as decorative, as it is present in sculptured form. And it is present in so cultured form that it helps in protection. So it acts as an armor, articulated. It is highly sculptured plates made up of cellulose and pectin. And these provide protections. So as these are sculptured plates, they provide protection. So they act as armor. They act as armor in protections. So let's move. So these are the sculptured plates. These are the sculptured plates. These are the called as sculptured plates. These are the sculptured plates present. And here what happened? The body is what now differentiated into a groove present here. And this group is called as transverse groove. This groove is called as transverse groove. And here we find another groove present and this groove is called as longitudinal groove. Longitudinal groove. So groove means depression. So from the transverse groove we find a flagella present and from the longitudinal groove we find a flagella present. So two different type of flagella are found to be present. One from the transverse depression and one from the longitudinal depression. So these dinoflagellate contains two different types of depressions, and on the depressions, above the depression, the body contains these sculptured plates. These are the plates, and they act as armored like structure, and these are highly sculptured. These are highly sculptured in nature. Got it? Now let's move. So we have discussed that these dinoflagellates do contain sculptured plates, and they have two types of groups, and we do call them as armored dinoflagellates because these sculptured plates provide protections from enemies, and also they contains the thecas have two groups, means they have two depressions. One depression is longitudinal presence, another depression is horizontally present. So two depressions are found to be present, and this longitudinal depression is called as sulcus. This longitudinal depression is called as sulcus, and this is called as annulus. The transverse depression is called as annulus, and the longitudinal depression is called as sulcus. And we find the other group's transverse is otherwise also called as cingulum or annulus or girdle. As we have discussed, this is a transverse group, so you can call different name to the transverse group. But longitudinal group is called as sulcus. This is the longitudinal group that is called as sulcus. But the transverse groups are sometimes called as cingulum, annulus or girdle. And as we know from this group, what happens? The flagella originates. So one of the flagella comes from the transverse group, another flagella comes from the longitudinal groups. So two different types of flagella present. One longitudinal arranged, one transverse. The longitudinal flagella is a thread-like structure. Is a thread-like flagella, whereas the transverse flagella is a ribbon-like nature. The transverse is a ribbon-like nature and is a thread-like nature, for which it is called as heterocont, because two different flagella are found to be present. Due to the presence of two different types of flagella in the same organism, it is called as heterocont flagella. And as two flagella present, so we call the organism as dinoflagellates. Now let's move to the next point. So as we know that the longitudinal groove and the transverse groove present, they contain small openings are present. So in the groups, in particular groups, small openings are present, and these openings are called as lorica. These openings are called as lorica. So two different lorica present, one on the transverse side and one on the longitudinal sides. Now what happens? From the transverse side, the flagella originates. From the longitudinal sides also the flagella originates. So two different types of flagella comes out. One from the longitudinal lorica, another one from a transverse lorica. But the transverse lorica flagella is a ribbon-like flagella. Is a ribbon-like, and the longitudinal one is a thread-like flagella. Is a thread-like. So this is how the flagella is being arranged. Now let's move that. What happens? That the transverse flagella lies in the circular groove, yes, and the longitudinal flagella lies in the longitudinal groove. As we go to the diagram, here we find what? Now we came to know that this particular nature is this is the transverse part. This part is the transverse part. So this transverse part must contain the transverse flagella, and this is the longitudinal flagella present. When we move into anatomy, we find this part is the transverse part, this part. So here the transverse flagella is present. And from here the longitudinal flagella is present. Inside we find the cell organ Golgi apparatus, we find mitochondria present, we find chloroplast present, we find vacuoles are present, and we find nucleus present at the center, and we find vesicles are present. These are different types of vesicles. So all eukaryotic organelles are found to be present. 70S ribosome is present, and very important, they also contain an iSpot is present. In some dinoflagellates, an iSpot-like structure is found to be present. Got it? So this is about the anatomy of this dinoflagellates. So we know very well the longitudinal flagella that originates is a very narrow, they're very smooth, and they are directed downward posteriorly. Whereas the flagella present, that is transverse flagella, they are ribbon shaped and they are arranged in circular manner. So this is the circular manner arrangement of transverse flagella, and this is the ribbon shaped. So both the flagella are 90 degree to each other. So both the flagella are always 90 degree to each other. Yes, the transverse flagella is a ribbon-like structure. and both are oriented 90 degree to each other and each other producing a spinning movement. Yes, the transverse flagella helps in spinning movement and the longitudinal flagella helps in body balance. So one helps in spinning movement, the ribbon shape, that is transverse one, another helps in, that is body balance. So by this nature, the spinning movement of this particular dinoflagellate is called as whirling whips. So they are called as whirling whips, the spinning nature of the dinoflagellates. Now let's move into its body coloration. We find different type of body color: brown, green, yellow, red, white. Now due to the presence of special type of pigments that is called as chromatophores that provide colorations. And these colorations are either due to chlorophyll A that do helps in photosynthesis, and they also contains beta carotene and also they contains alpha carotene. In addition to this carotene, they contains body coloration that is called as xanthophylls, and these xanthophylls contain different types of fucoxanthin. They contains different types of fucoxanthins, and these fucoxanthins combination they provide what? Now they provide the body color. Got it? And the best example where we find maximum amount of this xanthophylls is present, that is called as pyridinin. Now the plastids are generally surrounded by three membrane alveoli. Yes, the outer chloroplast membrane, this is the inner chloroplast membrane, and we have thylakoid membranes. Got it? And we have thylakoid membranes. So three membranes are present plastid. So plastid are three membrane envelope. So these three thylakoid lamella also present. Now we find inside the plastids we find lamella, and these are three thylakoid like structures present. So three thylakoid membrane placenta are found to be present. They, due to the presence of plastid, they are autotrophic and photosynthetic in nature. The best example is called as ceratium. And some are depends on dead organism and some on living organism. Very few are saprobiotic and very few are parasitic disease causing, but mostly they are photosynthetic or autotrophic in nature. The reserve food materials found in these dinoflagellates are carbohydrates and oils, and we find the nucleus is present at the center and very large in nature. Got it? Your nucleus present very large in nature, but the vacuoles present are in the form of contractile or non-contractile vacuoles. Yes, here we also find vacuoles, but the vacuoles are not present at the center and they are not very large as nucleus. But the vacuoles are of different types, either mostly non-contractile, some are having contractile vacuoles. Two different types of vacuoles are present. Okay. It has condensed chromosome even in the interphase. We know very well during cell division, in interphase the chromosome starts to release out. These are the thread-like structure. But in interphase we find the chromosomes are very condensed. As in the interphase which should be chromatin-like nature, but here the chromosomes are highly condensed because these are dinoflagellates are mesokaryons. We'll discuss what are mesokaryons. So these are the dinoflagellate eukaryotic cells. They don't have histone proteins, but they do have cell membranes, they do have cell organelles, they do have membrane bound organelles, they have nuclear membranes. So without histone proteins, but they have membrane bond organelles and a nuclear membrane, for which they are called as mesokaryon. Got it? So this is what we came to know. Yes, nuclear envelope, they contain nucleolus. Yes, they do contain the nucleolus, which is present also during cell division. So in all mesokaryons we find during cell division the nuclear membrane nucleolus are present, spindle fibers also present. So the remaining of this nuclear membrane, remaining of the nucleolus indicates that these are mesokaryons type of cell divisions. Clear? Got it? So dinoflagellates are type of best example. Yes, they are mesokaryons. These are called mesokaryons because they don't have histone proteins, but they do have eukaryotic nature. It was given a dog in 1966. Non-contractile vacuoles are present, and the non-contractile vacuoles present, these non-contractile vacuoles are present, these are called as pusule. Got it? These are non-contractile vacuoles present called as pustules. And they also contains one or two vesicles are present. So below non-contractile vacuoles, we find vesicles are found to be present below. And also we find contractile vacuoles present that helps in osmoregulation. These helps in non-contractile. So non-contractile vacuoles. These are non-contractile vacuoles and these are contractile vacuoles. The contractile vacuoles maintains concentration gradient, that is osmoregulation, whereas the non-contractile vacuoles helps in flotation. Some dinoflagellates contains stinging cells, a trichocyst, a cnidoblast like structure. As we have discussed in Animal Kingdom chapter that in Hydra or Cnidoblast, they contain stinging cells like structure for paralyzing its prey and for defense. These are called a Cnidoblast. It stings because it contains poison, hypnotoxin. So this type of same type of trichocysts are also found to be present in the cell of dinoflagellates that do help in prevention from enemy. Do it helps in preventing from enemy? Yes. And let's move into reproduction. Mostly the common mode of reproduction is asexual, that is by interfusion cell division. Yes. And they also contains isogamous and anisogamous sexual reproduction. Isogamous where the gametes are equal in shape, anisogamous where the gametes are unequal in shape. So, but no oogamous. They don't have oogamous type. We know very well what is oogamous type. Oogamous type is the union between non-motile and motile. So the union between motile and non-motile is called as oogamous, but dinoflagellates don't contain this oogamous type of reproduction, sexual reproduction. And the best example of sexual reproduction is found to be present in Ceratium. And normally the life cycle zygotic meiosis. What is zygotic meiosis? Only haploid, all haploid organism undergo zygotic meiosis. Let's discuss how haploid organism. For example, suppose this is the haploid organism. This is haploid organism. When it divides by cell division, so mitosis. So mitosis, it produces the gametes. And we know where gametes are N. Yes. Now the gametes undergo fertilization. So when the gamete undergo fertilization, what we got? We got the zygote. And the zygote is 2N. Got it? But the parent is M. So the zygote must return back to 2N. So the division of zygote to produce the haploid is called as zygotic meiosis. So the divide to produce the parent haploid. And this is called as zygotic meiosis. So 2N converted to N. Got it? So zygotic meiosis is mostly found to be present to seen in haploid organism. It is not seen in diploid organism, because in diploid organism we find that is called as gametic meiosis. Got it? Now let's move in the next point. The gametic meiosis is also found to be present in the organism that is called as Noctiluca. Most of the zygotic meiosis are found in Ceratium and Gymnodinium. But Noctiluca is that particular organism of dinoflagellates which undergo gametic meiosis. Now let's move into facts. Mostly dinoflagellates are marine and the bioluminescence means they emit light in the sea. So in the sea they emit light due to the nature of phosphorus. So they are called as bioluminescence. They look like stars, twinkling stars on the surface of the sea. They looks very beautiful. And the best example of bioluminescence dinoflagellates are called as Noctiluca, Pyrodinium and Pyrocystis. Noctiluca, Pyrodinium and Pyrocystis. Because why? They contains phosphorus, and due to phosphorus, phosphorescence takes place, and due to phosphorescence they emit light, for which the sea glow at night. Now some dinoflagellates are very poisonous. Goniolux catenella is that type of dinoflagellate that produce toxins, and the toxin name is called saxitoxin, and this saxitoxin paralyzes or causes death to the aquatic animals. So it causes death to aquatic animals. So these causes death to the aquatic animals. Got it? So these toxins are very poisonous to vertebrates, fish and other aquatic animals. But one of the organisms that is called as marine shellfish. This marine shellfish accumulates all the poison. They accumulate all the poison without any death. So it is only shellfish that can digest dinoflagellate without any death, because these poisons are harmless to this particular organism. So they are not harmful to this particular shellfish that is called as mussels. But the problem is that if human consume mussels, then human may face this disease, that is paralysis. Paralytic cell fish poisoning, PSP disease. So what is PSP disease? Paralytic cell fish poisoning. Because if human consume these mussels, and mussels contains the accumulation of poison, and these poison paralyze the nervous system. So as they paralyze the nervous system, it causes disease that is called as PSP. So they paralyze the humans. Some dinoflagellates, they multiply very rapidly. As they multiply very rapidly, they causes red tide. So this red tide is due to the proliferation of some dinoflagellates like goniologs and gymnodinium. So goniologs and gymnodinium are two different types of dinoflagellates that causes red tide due to maximum proliferations. Now let's discuss about practice some questions. Assertion: dinoflagellates often exhibit, yes, true. The pauses, yes, true. Yes, both are true. Got it? Next questions. Dinoflagellates biochemical mediated by specific enzymes called substrate, yes. The Lucifer, yes, both are true. The third question. Dinoflagellates are often responsible for red tides. Yes, it's true. Why? Feral condition, the explores releasing pigments and toxin to seawater. Siliceous in nature. Yes, this one is the right answer. Go through the answer. When certain dinoflagellates multiply rapidly, marine ecosystems suffer massive losses. Yes, because the red tides and toxins are produced. So as red tides and toxins are produced, definitely the losses. Blooms related toxins lethal to fish. Yes. Okay, the first one is correct. Now the fifth one, Biology Photosynthetic Protista is, that is dinoflagellates. Yes. So only 5 questions we have discussed, but you will get 1000 plus question per chapter in our site, that is called as this particular site, studyBioKaare.com. You can get maximum question. If you want for online practice to know your score at particular time, you can go for it. If you want any doubt clearing classes, you can also go for doubt clearing class in this particular site. And if you want direct PDF, direct mind maps, worksheets, MCQs, you can directly go to this particular site and download it. And this is a particular number you can directly contact for any doubt, or you can go to our channel, visit our channel and download all the study materials, PDF, etc. etc. So this is all about the dinoflagellates, general characteristics, life cycles, and the examples, its facts, its economic importance, and harmful aspects, everything we have discussed. So in the next class, we'll go to and discuss about, that is called as the next photosynthetic protist, that is Euglena. Okay. Hope you all enjoyed. Thank you. Okay. Today we are going to start a new photosynthetic example, that is called as Euglena or Euglenoids. It is also a photosynthetic protista. So we have already discussed diatoms. We have already discussed dinoflagellates. So today we are going to discuss the last photosynthetic protista, that is called as Euglenoids. Let's start. So these Euglenoids are group of chlorophyllous, means they contain chlorophyll. So they are chlorophyllous. And also they don't contain chlorophylls, chlorophyllous flagellated plant. Means these are the groups where we find chlorophyllous group present and non-chlorophyllous groups are present, and they do contain flagella. So here it indicates that they behave as plant plus animal nature. So Euglena contains plant character as well as animal character both. And it is regarded as the largest genera. Euglena is regarded as the largest genera. Euglena is the largest genera among euglenoids. Now these euglenoids are normally unicellular, they are flagellated, and they are mostly present in the fresh water, that is stagnant fresh water or damp soil. So normally when we find Euglena, they are flagellated, yes. They are unicellular, yes. And normally they are present in moisture-based soil, or it is found in stagnant water, that is mostly fresh water. So majority of them are fresh water, yes. They are fresh water. And most of them are found to be present in stagnant water. And the body is spindle shaped, yes. The body is spindle shaped like structure. The body is spindle shaped with blunt anterior end and pointed posterior end. The posterior end is very pointed, but the anterior end is blunt. So in the anterior end, we'll find this type of animal-like nature, and the posterior end is pointed. Now this one is pointed. So this is the posterior part. This is pointed, and this are the anterior part. This is blunt. These are the blunt-like appearance. Got it? So they have spindle-shaped body. So both the end pointed, and the middle part is wider. So this nature is called a spindle shape with blunt anterior. So anterior end is little blunt, little wider, not so pointed, but the posterior end is pointed. Why it is not pointed? Because it is differentiated into animal-like nature. You will find the animal character, the mouth present, the pharynx present, the reservoir present. We'll discuss later. Cell wall is totally absent. Yes, cell wall is absent. But the body is covered by a covering. The body is covered by a elastic protein covering, and this covering is called as pellicle. They don't have cell wall. Yes, they don't have cell wall. But the body is covered by a protective covering that is called as pellicle, which is protein in nature and highly elastic in nature. So periplast, this pellicle is otherwise called as periplast, which is protein in nature and they are highly elastic. As we have discussed now, suppose this is the body. So outside the body we find a covering present. This is not cell wall. It is called as the pellicle. And this pellicle is protein in nature and highly protective and highly elastic in nature. Got it? Now let's move. The locomotor organs is flagella. Yes, they do contain flagella. So flagella is the main locomotor organ is present. And the cell bears a long flagella, which is a long thin cell. And we do call it as trichonematic, arising at the anterior end, and a short flagella. For example, suppose this is the body of Euglena, for example. So it contains a long flagella, which is parallel with the body. And a short flagella is also present, which is inside the body, blunted. So this is thin cell, means it is smooth. If any flagella that is smooth, they don't have hair in nature, they are called as thin cell. So it is a thin cell type flagellum present. Then we find actually there are two flagella, but one of these is highly reduced. For example, suppose this is the mouth part, so the anterior part. So one of the flagella, they moves out, but other flagella present which is very short, they never comes out of the body. So two flagella present, one coming out of the body, one present within the body. Got it? So they are very reduced in nature. And the longer flagella has two branches. Yes, they have two branches. Longer flagella have two branches. The two branches with two basal bodies or basal granules or blepharoplast. So the longer flagella contains two branches. And each branch has its own basal granules. Yes, as the flagella bifurcates, and this is the flagella for it. So each flagella contains its own basal granules. These are the basal granules present, and these are the branches present of the flagella. So this particular flagella is a flagella that do helps in its locomotion. Okay. For example, suppose this is Euglena present. Got it? And here we find that the flagella comes out. The meeting point of flagella. Here the meeting point of flagella. So the union of two flagella is present. Parallel to it, we find a body present, and this body present at the meeting point is flagella is called as paraflagellar body. Paraflagellar body. Got it? So when the flagella meets the meeting point of flagella and just the body, so in this nature we find a body present, and this particular body is called as paraflagellar body. So at the meeting point of the flagella we find a special photosensitive body present, and this photosensitive body is called as paraflagellar body. It's very important point. Now, myonemes are present as oblique parallel. For example, suppose this is the body present, and around body we find the pellicles are present. For example, this is the pellicles are present. Got it? And this is the inside. Suppose body is present. So body is covered by pellicle. Here what happens? In addition to pellicle, from this pellicle nature, from this pellicle to this pellicle, we find that oblique fibrous-like structures are present. And these oblique fiber-like structures are called as myonemes. So these are called as myonemes. Got it? So these are oblique, parallelly oblique arranged stripes are present, which are attached from one pellicle of other side to the other pellicle, and these oblique stripes present parallel to each other is called as myonemes. And euglenoids forms creeping movement, means suppose this is a surface, so they move by creeping. They creeps and moves in the mode, and this type of movement is due to contraction and relaxation. So creeping movement is only possible by contracting and again relaxing. Again contracting, again relaxing. So this type of movement is called as creeping movement, and this is the contraction and expansion of this particular organism. Let's discuss about the structure. For example, this is the upper part present. This is the upper part present. This is the upper part present. And here what find? we find it is the animal nature. This is the animal part, and the lower part is the plant part. The upper part is the animal part and the lower part is the plant part. In the upper part we find that this is the flagella present, and outside the body covering we find the pellicle is present here. We find oblique steps are present. These are called as myonym is present. Inside we find the nucleus is present. Here we find the chloroplast are present. Paraphyllum bodies are present. But here what happened, the antermost end is called as the mouth. This particular gap is called as the gut, or the cytopharynx called as pharynx. And here the meeting point is called as paraflagella body present at the meeting point. And next to the meeting point we find a eye spot is present, that is called a stigma. Stigma or eye spot is present. Not it. And here we find a swollen region present at the bottom region of this body, and this is called as the reservoir. This is the reservoir of the stomach, is called as the stomach of the body. And also above below this reservoir we find below that contractile vacuoles are present for osmoregulations. And in the down part we find the plant-like nature, because chloroplasts are present in the lower part. The upper part behaves animal-like nature and the lower part behaves plant-like nature. This is what the structure means. So this movement, the creeping movement, is sometimes called as metaboly movement or euglenoid movement. So this creeping movement, this creeping type of movement is called as metaboly movement, or it is sometimes called as euglenoid movement. The apical end of the cell bears an invagination with 3 distinct parts. Yes, let's move what? Suppose this is the apical part present. So the apical end, when we move on to the apical end, we find 3 parts. Here is the mouth part present, here is the pharynx part present, and here is the reservoir present. So 3 distinct parts present like animal nature. The opening part is called as the mouth. Here is the mouth present. And this cavity, this canal is called as the pharynx. This canal is called as the pharynx. And here we find the reservoir present. The stomach-like activity is present, that is called as the reservoir. This is called as the reservoir present. So this particular part, the upper part of the body of the Euglena, they behaves as animal nature, and here also we find what? This flagella is present, and here we find paraflagella body, and the side of the paraflagella body we find a eye present, or eye spot present, that is called as stigma. We find a stigma present parallel to the paraflagella body, and this is called as near to the reservoir membrane, and this is called as the stigma. Is the eye spot. We'll discuss later. Okay, we find the reservoir helps in ingestion of solid food particles. Got it? So this particular part, the reservoir helps in ingestion. The food is engulfed, taken into this transport through, and here it is present and it moves down. Here we find contractile vacuoles, food vacuoles are present. So these food vacuoles will digest this particular food and then distribute throughout the cell. So this is how it is behaves as the animal nature, the upper part. Now, stigma and eye spot is present, just how we discussed. Suppose this is the upper part present. Suppose the upper part of the body present. And suppose this is the paraflagella body present. So here is the paraflagella body. Next to paraflagella body, we find the eye spot present. And this is called as the stigma or the eye spot. It is photosensitive. It is very photosensitive. So they can feel light, they can sense the light, because they are photosensitive in nature. Yes, it is present in paraflagella body, just behind the paraflagella body. Stigma along with seems to be light sensitive, and it perception of light and stimulus light. It is photosensitive to red, orange light. And the contains a pigment that is called as astaxanthin. So astaxanthin is the pigment present in the eye spot. And this pigment is very sensitive to red and orange light. Red and orange wavelength of the light is sensitive only due to this particular pigment present in the eye spot, that is very highly photosensitive in nature. It also contains contractile vacuoles, and it helps in osmoregulation. It helps osmoregulation. And also it is present just below the reservoir, just we have discussed. Suppose this is the reservoir present. So below reservoir we find the contractile vacuoles present, because this is the reservoir and this is the contractile vacuoles. The contractile vacuoles helps in osmoregulation and excretion, that is to maintain chemical balance in the body. Single large nucleus lies at the center. We know very well. Suppose this is the blunt end. This is the blunt end as we have discussed. And this is the pointed end. Yes, as we have discussed. In this nature, here we find the nucleus present. So here we find the nucleus present because the upper part behaves as animal nature. The upper part is animal nature, whereas the lower part behaves as plant-like nature. This is the plant-like nature. And the nutrition is autotrophic because they contains chloroplast. Yes, they contains chloroplast. Here the chloroplast are present. So Euglena viridis is a photosynthetic. However, some are saprobiotic. Means they depends on dead and decay. But mostly they are photo. But some are saprobiotic. Means they depend on dead organic matter. So they get nourishment by secreting the digestive enzymes in the absence of light. So if light is absent, then they undergo animal behavior and they contain the digestive enzymes to digest the food materials. If light is present, then digestive enzyme is reduced and with help of light they prepare their own food. So they do have both plant plus animal nature. So Euglena have both plant nature and animal nature. So this dual mode of nutrition in Euglena is called as mixotropic mode of nutrition. Yes, mixotropic means dual nutrition, two types of nutrition. One is photosynthetic, another one is saprobiotic. Got it? So holozoic nutrition is absent. So direct engulfing, that as we ingest the food or as we intake the food, so we intake the food in holozoic form. So this type of nutrition is absent in this Euglena. Some, very few of them have holozoic. That is, the best example is called as Paranema. Paranema is one organism of Euglena where we find holozoic type of movement digestion present, where the food is engulfed, ingested. Most of them are, some of them are saprobiotic. That is Rhabdomonas. Rhabdomonas is the best example of saprobiotic in nature. So Rhabdomonas is saprobiotic. Photosynthetically, they hence due to the presence of chlorophyll A, so they prepare their own food. They do also contain chlorophyll B. So this two chlorophyll behaves as plant nature. So higher plants contains chlorophyll A and chlorophyll B to prepare their own food. Same thing, this Euglena contains chlorophyll A and chlorophyll B to prepare their own food. Yes. So this photosynthetic pigments also present, that is called xanthophyll, to provide body coloration. As in other photosynthetic protista, we have discussed xanthophyll, fucoxanthin, diatoxanthins, diadenoxanthins. Here also we find xanthophylls are found to be present to provide body colorations. They contains beta carotenes. But in dinoflagellate we find alpha and beta carotenes are both present, but here only beta carotenes are present. In addition to other pigments, beta carotene is also present. This is a very important point. The reserve food materials is a type of polysaccharide that is called as paramylon. The reserve food material is paramylon. So the stored cytoplasm in the form of paramylon granules means inside the body the food materials are stored in the form of these granule-like forms, and these granular forms are called as paramylon forms. And these are the paramylons that helps in storing the food materials in the form of carbohydrate or starch. And they are chemically beta-1,3 glucans. Yes, glucans is polysaccharide. So normally paramylons are what? These are beta-1,3 glucans. Very important. So paramylons are beta-1,3 glucans. Now under favorable conditions, they divide by longitudinal binary fission. Very important question. So under favorable condition, they mainly reproduce by longitudinal binary fissions. Got it? Very, very important point. But under unfavorable conditions, they go for palmella stage or cyst formation. They undergo cyst or palmella stage like conditions, where they undergo perination. Means they, under unfavorable condition, they just becomes dormant to withstand this adverse condition. So when condition favors, then they again reproduce. Sexual reproduction is not present and not known at present. It's not found. And the best example Euglena is called is Euglena and Euglena. The Euglenoids and Euglenoids we find Paranema and Euglena are the best examples. Now let's move into different facts. Euglena is both producer and decomposer because they are photosynthetic and they are saprobiotic. So they are producer and decomposer both. It is studied that they are plant as well as animals. Yes, we have discussed. So it is sometimes called as plant animal. It is sometimes called as plant animal. And also the plant characters are Euglena due to the presence of chloroplast and pigments. Why it is plant character? Because the presence of chloroplast and pigment, chlorophyll A, chlorophyll B, beta carotenes, xanthophylls are present. And plant characters of Euglena include holophytic nutrition, that is autotrophic mode of nutrition. But animal type, they don't have cell wall. They do have a protein covering that is called as pellicle. And they have eyes present, stigma, and a paraflagella body is present, which is animal character. And also the contains contractile vacuole for osmoregulation, and that is called as excretion. So which is an animal character in nature. And also animals are character, they go longitudinal binary fissions. They go longitudinally binary fissions. Now let's go for some practice questions. First example is Euglena is called a kinetic link between plants and animals. Yes, right. It posts chloroplast photosynthesis and absence of. Yes, both are right. Yes, this is the right answer and this is the right explanation. Now let's consider the following statement regarding Euglena. Let's see. It has protein rich covering called pellicle. Yes. It possesses two unequal flagella. Yes. Its reserve food material is paramylum. Yes. The cell wall is made up of cellulose. They don't have cell wall. They don't have cell wall. Choose the correct option. 1, 2, and 3. Yes. 1, 2, or 3 are right, or others are wrong. Got it clear? Third question. The flexible movement of Euglena is mainly due to the presence of myonemes. Cell wall? No. Silica cell? No. Pellicle present? Yes. Pellicle contains myonemes. And due to the presence of pellicle and myonemes, the euglenoid movement is only possible. Got it. Next questions. Which one of the following there is a food materials? That is paramylon, starch, glycogen, paramylon, laminarose. So it is not, it is not, it is not. So paramylon is the right answer. Why is Euglena considered as mixotropist? Both type of nutrition is found to be present. It can perform photosynthesis, also heterotroph. So this one is right. It has both plant and animal cells. Okay, for nutrition question, major nutrition, this one is the right answer. Alright. So only 5 questions we have discussed. You can go for 1000 plus questions in this particular site. You can give the online exam and practice your score at particular time, or you can go for the clearing classes here. If you want direct PDF or you want mind maps, worksheets, MCQ, direct MCQ PDFs, you can go to this particular site. Or if you have any doubt you want to clear it out personally, you can go through this number. Or if you have any, if you want to download ample amount of questions and PDF, then you can go for this particular channels. So this is all about of Euglena. That's the photosynthetic protista we have discussed. So we have completed photosynthetic protista. Last class we have discussed dinoflagellates, and before that we have already discussed diatoms. So we have completed photosynthetic protista. Okay, thank you. Hope you all enjoyed. Okay, today we will discuss about that is the protista, that is the decomposer protista, or we do call them as consumer protista. So we have already completed photosynthetic protista, and is the second group of protista that is called as the decomposer protista, fungi protista, and the best example is called as slime molds are called the best examples. So these are decomposer protista, the slime molds present. So they include in the class that is called as Mycomycetes. So these slime molds are included in the class that is called as Mycomycetes of fungi. Got it? And they were also called Mycetozoa by the fellow that is called as the father of fungi, that is called D. Berry. D. Berry called them as Mycetozoa because they also contains animal-like nature, fungal-like nature and animal-like nature. Due to close related to animals, these one type fungi related with animals, so they call them as that is Mycetozoa. And mycologists sometimes include them, some mycologists include them as Gymnomycota because they also looks like gymnosperm like nature. So this is the slime mold, and this is the small nucleus are present within it, and this is the plasmodium structure. This is plasmodium like structure. So these slime molds are called as protistian fungi. The slime molds are called as protistian fungi. Let's move into general character as we are going to discuss two types. One is called as acellular slime molds. We will discuss acellular slime molds, and again we will discuss about cellular slime molds. This is acellular slime mold because they don't have cell. All the nucleus are present, multinucleated. So the multinuclear condition is called as plasmodium. We will discuss later. So let's move. So these are the free living organisms. Yes, They are creeping on dead debris, the dead debris of fallen leaves or rotten logs or wood. So they are saprophytic and they are decomposer because they always as creepers. They creep on dead debris or leaves, dead parts of the fallen leaves or trees. Got it? They have naked protoplast, means the body present here, and the body contains multinucleated. So nucleus is multinucleated present. These are the many nucleus present. The cell membrane present. No cell wall. No cell wall. At present they don't have the cell wall. So these are the nucleus and these are the nucleus. And this condition is called as plasmodium. This condition is called as plasmodium. So they are called as naked protoplast-like structure. And they are not covered by any cell wall. As I discussed, they are not covered by any cell wall in the vegetative stage. But in the reproductive stage, it is found. The cell wall is found. It is found in the reproductive stage. It is found in the reproductive stage. Yes, we'll discuss. It is found in the reproductive stage, but it is absent in the vegetative stage. And they also don't have chloroplast or chlorophyll. So they don't have chlorophyll. As they don't have chlorophyll, they are fungal in nature. These are fungi. They don't have chlorophyll. And they are saprobic, mode of nutrition. That means that depends on dead organic matter for the nutritions. And they are phagotropic. They are phagotropic, mode of nutrition. Phagocytes means they are phagocytes. They engulf the food materials directly. So intake of solid food materials means they intake solid materials. They intake solid food. The intake of solid food nature is called as phagocytic in nature. Means they engulf the food materials. Got it? Suppose this is the body present, so they engulf the food materials. And then what happens? Food materials is engulfed and taken into it and they can dissolve. So this type of engulfing the food materials is called as phagotropic type of nutrition or cell eating. Means they intake the solid food materials directly into the body. Under favorable conditions, they form aggregation called plasmodium. Means they form groups to form multinucleated structure. This is called as plasmodium. So the plasmodium may grow and spread to several feet. Yes, the plasmodium can grow and spread to several feet, and normally the plasmodiums are multinucleated in structure. These are the plasmodium, and they move into several feet. They can grow in all direction into several feet like structure. So during unfavorable condition, during favorable condition, they go for plasmodium. But when the condition is unfavorable, these plasmodium, they rest and they gives out fruiting bodies. So this plasmodium gives out fruiting bodies, and these are called as the fruiting bodies. And this is the plasmodium present. The plasmodium growth is restricted in unfavorable conditions, and they gives out fruiting bodies to continue its generations. And these fruiting bodies are sometimes called as sporocarp. This is called as sporocarp. Yes. So the fruiting bodies bear spores at the tips, and during the life cycle, they also bear non-cellulosic amoeboid-like cells. So in the life cycle, during the life cycle, they bear amoeboid-like cells, irregular like this type of cells are present. This is called amoeboid-like cells present in its life cycles, which they don't have they don't have cell wall, no cell wall. As we have discussed, means they are non-cellulosic. They don't have cell wall. And the spores have cell wall. Means the reproductive stage. The reproductive stage has cell wall. The reproductive stage has cell wall. But the vegetative cells don't have cell wall. This is a very important vital character of this particular slime molds. So the vegetative phase resembles that with animals, but the reproductive phase resembles that with plants. Got it? Because vegetative phase don't have cell wall, so they resembles with that of animals. The reproductive phase do have cell wall, so they resembles with plants. Yes. Amoeboid plasmodial stage resembles protozoa, and they also contain amoeboid type of structure. So this amoeboid structure resembles protozoa. So these slime molds resembles plant, resembles animals, and resembles protozoa. And also it is a fungi. It's a very confusing organism. Spore forming nature is due to fungi. Yes. So this contains fungi-like character. This contains plant-like character. This contains protozoa-like character. This contains animal-like character. So the spores producing organism, I'm having this stalk, and on the stalk the spores are produced as conidia and sporangia produced on the stalk. So like this is fungi nature. Spores are extremely resistant. They are highly resistant against adverse conditions. As in fungi, the spores can survive for many years and can survive in adverse conditions. And these spores are dispersed by air. Yes, normally the spores are dispersed by air currents. Very important questions. Reproduction is both asexual and sexual means in slimes. And we are talking about the general character of slime molds. Then we'll go for cellular type and acellular type. This group is represented by two separate types of organism. One is acellular, another one is called as cellular. Yes, one is called as acellular, means they don't have cellular, means the body is multinucleated. If the body is multinucleated, it is called as plasmodium. Acellular. If the body is uninucleated, it is called as cellular. So the both mode of organism is present in this particular group. Let's discuss acellular mode. So in acellular, they move on decaying leaves and lumber, and the slimy masses means they secrete. This is a plasmodium present. They move on any organism and they secrete a mucilus slimy, and through the slimy they creep. They creep on the organism and they dissolve the organism. They release the digestive enzymes and dissolves the organism and intake the materials, dissolved materials. So somatic body is free living, yes, multinucleated plasmodium nature. This is a multinucleated, and the nucleus is 2N. The nucleus is 2N. It is a diploid set of organism. So acellular slime molds sometimes called as plasmodial slime molds. So acellular slime molds are also called as, that is called as plasmodial slime. So acellular slime molds called as plasmodial slime molds. They are slimy masses present on decaying leaves and lumber. So they creep on them, release the digestive enzymes, then dissolves the organism and intake the materials. And the somatic body is free living, multinucleated, called as plasmodium. The movement occurs by means of pseudopodia. Yes, they contain pseudopodia like amoeboid-like structure. So movement is due to a pseudopodia, the vegetative structure. But in the reproductive structure, we will find the flagella. Under unfilled conditions, they contain the fruiting bodies. So this plasmodium gives rise to fruiting bodies. So they give rise to this type of fruiting bodies present. And this is the plasmodium. Yes. So under unfavorable conditions, the plasmodium reduces and halt its growth. And so they give rise to fruiting bodies. So this fruiting body sporocarp, which produce the spores, is only possible in unfavorable condition. But in favorable condition, plasmodium undergo division, cell division and multiply. So the sporocarp contains a stalk, and the sporocarp contains sporangium at its tip, and this sporangium wall is called as peridium. For example, suppose this is the fruiting bodies. Got it? So this stalk is called as this stalk. Sporangiospore. Sporangiophore. Got it? So 4 is used for stalk. And this is the called as sporangia. This is called as sporangia. This is called as sporangia. The total is called as sporocarp. The total structure is sporocarp. Within sporangia, we find the spores are present. Within sporangia, we find the spores. But the sporangia structure, the sporangia is externally lined with this covering like structure, and this covering like structure is called as peridium. This is called as peridium. Got it? So peridium are the wall of the sporangia. The sporangia contains a specialized wall that is called as peridium, and these peridiums are present on the wall of sporangia. So the fruiting body is otherwise called as sporocarp, as we have discussed. Yes, here it is the sporocarp, and these are the lines present. The covering is called as peridium, and here we find the lines are present called as capitulum. So let's discuss what is capitulum and what is peridium. So this total covering is called as peridium. On the peridium, we find the lines are present. These lines are called as capitulum. And inside the body, we'll find the spores present, and in the stalks type, a sporocarp is found to be present. Got it? So sporangia has an intricate network of cytoplasmic threads called as capitulum. Yes. So here we'll find sporangia. So the sporangia present, the sporangia contains this thread-like structure on its wall. And this thread-like structure is called as capitulum, and the covering is called as peridium. And the covering is called as peridium. So this is the structure sporocarp. And the spore wall is double layer. The spore wall is always double layer. So in the reproductive stage, we find the spore wall, which is of double layer. Got it? Clear? And in some group we find the spore wall, the outer wall is spiny and sculptured. So the outer part of the wall is spiny and sculptured. The outer wall is spiny and sculptured. Got it? So when we move on the sporangium, spore wall spiny and sculptured, but the inner wall is very smooth. So this is the outer wall which is spiny and sculptured. Got it? And inside we find the spores are present. This is the structure. And the chief mode of nutrition is called as saprotrophic. Means as they are decomposer, so they are saprotrophic mode of nutrition, and they don't have the cell body because the cell body is multinucleated plasmodium. So it is multinucleated structure, so we call them acellular. So on germination of the spores, they give rise to biflagellated swarms or non-motile mixed amoeba. Suppose for example, this is the spore present. Got it? Sporangia present. So when the sporangia, when the spore falls down on the ground, when the condition is suitable, so one of the spore, they give rise to gametes, and these gametes form having flagella-like structure. So these gametes are present, and some other group spores differentiate to form amoeba-like structure. And these are the gametes. Got it? These are the gametes. So these gametes are called as swarm cell. These are called as swarm cells, and these are called as mixed amoeba. Got it? So what we find when the spore germinates, they give rise to two types of gametes. One is that is called as giving flagellated gametes, swarm cells. Another one is amoeboid gamete, that is called as mixed amoeba. Let's discuss one by one. So biflagellated swarm cells act as gametes. Yes, just now we discovered. So biflagellated swarm cells acts as gametes. And also non-motile mixed amoeba, the as mixed amoeba, this mixome, as we have discussed now. So sexual reproduction isogamous means the union of mixed amoeba, or the union of swarm cells, the union of mixed amoeba or the union of swarm cells, normally they are of this type. The plasmodium becomes multinucleated. Yes, the plasmodium becomes multinucleated, and this multinucleus plasmodium, the plasmodium become multinucleated, yes, and the reproduce undergo binary fission. So this plasmodium present, they undergo binary fission to give rise the daughter plasmodium. The best example of acellular type is called as Fusarium, Fusarella, and Fuligo. These are the best example of acellular slime mode. So we have completed acellular slime mode or plasmodial slime modes because it is multinucleated, they don't have cell-like structure. Let's discuss about its structure. For example, suppose we are taking this. So this is the young plasmodium. So this is young plasmodium because this is a multinucleated structure present. Got it? So this gives out, they germinate and they stretch, elongates. When the condition is unfavorable, they reduce its structure and they give fruiting bodies. These fruiting bodies, the formation of fruiting bodies called as fructification. And here we find the fruiting bodies are present, and these are the sporocarp fruiting bodies, sporangia. They contains capitulum present, the iridiums are present, and these fruiting bodies gives rise to spores, because as we know these are 2n, the plant body is 2n, we know the body is 2n, so they undergo meiosis. So undergo meiosis to produce n. Because the fruiting bodies all are 2n, they are inside the fruiting bodies, meiosis takes place. And when the spores germinate, they germinate two types. One is a mixed amoeba type, amoeba-like structure, and one is a biflagellated, so one small flagella, one long flagella, swarm cells. These are called as swarm cells. And both they act as gamete. This is a gamete, either they will form this type or they will form this type, it depends. So most of them forms of this type. And this mixed amoeba, they go for union, and this type of union is called isogamy. So in isogamy, initially the protoplasm takes place, union is called as plasmogamy. Union of protoplasm is called as plasmogamy. Then the nucleus union is called as karyogamy. So when the nucleus unite, they form zygote 2n, and this 2n germinates to give that is called as plasmodium. So they contains diplophase and haplophase present. So this is the acellular mode of nutrition. Life cycle, that is of acellular life cycle. Now let's discuss about cellular slime modes. Means they contains mixed amoeba-like cell-like structure. So they have unicellular cell-like structure. So these slime molds are cellular slime molds, also called as communal slime molds. What is communal? The colonial. These are colonial. They form colonies. They form colonies, pseudofilaments. They form small filaments, pseudofilaments or pseudoplasmodium, because they have the power of formation of colonies. And they don't have wall. Yes, they don't have wall. They are uninucleated and they are called as mixed amoeba. So they are mixed amoeba-like structure, uninucleated. So this is mixed amoeba. No wall is present. They don't have cell wall. So no wall is present. And complete absence of flagella. They don't have flagella in its life cycle. Very important question. Means in cellular slime molds, no flagella in its life cycle. No flagella is found to be present in its life cycles. Sporangia present and naked means they don't have no cell wall. So here cell wall, they don't have sporangia. We find sporangia just like present. And this is the covering, just covering of sporangia. And here we find the spores are present inside the sporangia. And spores are cellulose. When the spore release, and these spores, the spores are cellulose. It means sporangia is non-cellular, but the spores are cellulose because they have what? Cell wall present. And here it is called as the cell membrane. Got it? And this part is called as the spore. So the spore contains cell wall, but the sporangia don't have the cell wall. Sexually reproduced by anisogamous means one of the mixed amoeba, another of the mixed amoeba. So both are unequal in size. So this type of union is called as anisogamy. And we find the common cellular slime mold is called as dictyostela. It's a very important example, and which is a colonial forms, means they form colonies. So as they form colonies, they looks like pseudofilaments. Actually, they are not pseudoplasmodium, they are colonial form. They contain hundreds of uninucleated haploid flagella. So these cellular are haploid. These cellular are haploid type, but non-cellular are diploid type. So these haploid type of colonial forms are found to be present. So these aggregation forms colonies. So these are the aggregation of cells that they form colonies, fuse, and the colony gives the appearance. Actually, it is not multinucleated, but it looks like it is multinucleated, but it is truly not multinucleated. It is truly not multinucleated, but it looks like multinucleated as they form colonies. So sometimes we call them as pseudoplasmodium because they look like multinucleated, but actually they are not multinucleated. So they look like multinucleated, but they are not multinucleated. So sometimes called as pseudoplasmodium, we call them as pseudoplasmodium. So under adverse conditions, when the food supply is exhausted, at that time they contain cyclic AMP and they release a chemical that is called acrosine. So during deficiency of food, all the uninucleated, they come and gets aggregated to form colony to survive from adverse condition because the food matrix is less. So this aggregation is due to this particular chemical that is called as accretion, and is due to the presence of this particular cyclic AMP nature chemical reaction. So during adverse conditions, when food supply is less, when the food supply is less, then they go for this colonial form, aggregation form. But when the food supply is maximum, then they are separated and they can survive in along. Okay, now this type of movement is called as chemotactic movement during the formation of pseudomodium because the accretion chemicals helps them to aggregate. So these chemicals helps them for aggregation. So this type of movement towards each other is called as chemotactic movement found in cellular slime molds. Very important questions. So pseudoplasmodium exhibits primitive form of multicellularity. Means here just starting of multicellularity starts because already unicellular, so we are discussing unicellular. But multicellular nature starts from this. So we could sometimes call them as communal form, communal slime molds. So primitive multicellularity structure starts with your tissue system will start, that is called as division of labor. So it is the initial stage. So for which we do call them as communal slime molds because we find this colonial type, or that is this cellular slime mold is regarded as the advanced type of protista, or it is the primitive fungi but advanced protista. Got it? Clear? Because they contains communal nature, because from this particular slime, cellular slime molds, tissue system starts evolving. So the being of tissue system is only from this particular organism. During unfavorable conditions, they form cyst, and the formation of cyst is called as microcystis. For example, this is mixed amoeba. For example, this is mixed amoeba. Suppose condition is adverse. So when the condition is adverse, So when the condition is adverse, they produce a cyst, and this is the cyst. And this formation of cyst together organism is called as microcyst. Got it? Without cyst is called as mixed amoeba. Yes, called as mixed amoeba. But when the cyst is formed due to unfavorable condition, this together is named as microcyst. Got it clear? Now let's discuss. Microcyst is formed by perennation. Yes, why microcysts are formed? The microcysts are formed by perennation, means they will withstand adverse conditions, and by dispersals nature. Let's move. So we have discussed that this particular nature of slime multicellular is the advanced nature. Under dry condition, the pseudoplasm produces stalk spore. When the condition is dry, the pseudoplasmodium present, they gives rise to sporocarp. They again gives rise to sporocarp. Got it? So this pseudoplasmodium, this is called as the colonial form. This is the pseudoplasmodium. So this pseudoplasmodium gives the sporocarp of the fruiting bodies in adverse conditions. The sporocarp may be branched or unbranched. Yes, the sporocarp may be branched or the sporocarp is unbranched. It depends upon the nature of the sporocarp. Each branch bears single sporangium, as we have discussed. Each branch bears single sporangium. So this is the sporangium, this is the sporangium terminally, and these are monocentric. Yes, once is present. Sporangia don't have wall. No wall is present, no cell wall. But in cellular form, cell wall is found to be present like peridium. But in cellular form, there is no cell wall present in sporangia, but the cell wall contains cell wall. Within sporangium, we find amoeboid cells are present within sporangium, and these spore walls are present around it. On the approach of favorable condition, these spores liberate and germinate into a new organism. Sexual reproduction is also anisogamous, as we have discussed. One of them is bigger one is smaller one, and that union is called as an isogamous type of union, as we have discussed earlier. So during sexual reproduction, number of mixed amoeba form a clump. For examples of this mixed amoeba, this is mixed amoeba. So all of the mixed amoeba, they form a clump, and all the clump aggregates to form a single bigger organism. Got it? They all aggregate to form a single bigger, because they all engulf to each other and form a large one with smaller engulfing mixed amoeba. And this plasmogamy, and then plasmogamy, inside plasmogamy occurs, means union of protoplasm. The union of protoplasm inside takes place because all the mixed amoeba are engulfed within a single bigger one, and this bigger one dissolves all the smaller one within it. So all the protoplasm gets union, and this nature is called as plasmogamy. After plasmogamy, karyogamy takes place. Fused protoplasm secreted thick wall around it, and karyogamy takes place. And this thick wall forms microcyst. So this fused protoplasm, they produce a thick wall, and this thick wall is called as microcyst. So the macrocyst undergoes karyogamy. So normally what happens? This from the macrocyst present karyogamy, means the union of protoplasm takes place, the union of nucleus takes place. After the union of nucleus takes place, they become diploid zygote. And this diploid zygote undergoes meiosis to produce haploid, because the parent body is haploid. So zygote divides to produce meiosis. So this type of division is called as zygotic meiosis. It is called as zygotic meiosis. So this type of division is called as zygotic meiosis, as we came to know. So several mitotic division then occurs after that, and that gives rise to the mixed amoeba. Haploid mixed amoeba is formed, as the parent type is formed. Got it? And they release and rupture, and macrocystic wall gives out. Germination takes place. The best example is called a dictyostelium. Is the best example. Some examples are polyspondylia. These are the two best examples, but the most example, the common example that comes in the exam, that is called as dictyostelium. So slime molds, we can justify the protista forms having a connecting link, yes, between plants, between animals and fungi. It is very important questions. So slime molds forms a connecting link between plants, between animals and between fungi. Got it? And fungi-like feature means they have fruiting body sporocarp. They have fruiting body is called a sporocarp. Plants like means they are spore having cell wall. Cell wall. And animal like nature means plasmodium. They have multinucleated. As multinucleated, so they are plasmodium nature. So let's discuss about this particular life cycle. So let's see. So here we find the actual mixed amoeba present, single cell. They divide mitotically repeatably, it is haploid. This is haploid. And they form many amoeboid cells like structure. When condition is unfavorable, they aggregate themselves with the help of accretion chemical, cyclic AMP. And this aggregation is called as communal type or colonial type, or is called as pseudoplasmodium, this aggregation. After that, what happens? They gives out this plasmodium, they form a plasmodium-like structure. And this plasmodium gives like the sporangial structure for fruiting bodies. And these fruiting bodies produce the spores, it is called as N. And this N germinates to produce. So this is the life cycle of this particular organism that is called a dictyostelium. Got it clear? Now let's discuss. We have completed. Now let's go for some practice questions. Slime molds are placed on the Kingdom Protista. Yes. During the vegetative phase, they exhibit as amoeboid mass or without a cell wall. Yes, yes, yes. Both A and R are true. Then we move on to consider the following statement regarding the slime molds. They are saprophytic. Yes. Under favorable condition, they form an amoeboid plasmodium. Yes. The spores possess two flagella. False. The spores are highly resistant and scalpel. Yes, true. So the correct option is 1, 2, and 4. These are not the correct option. The third question, the feeding stage of slime molds is known as mycelium, no. Plasmodium, no. Sporocarp, no. The plasmodium, because they engulf the matter. The feeding mode is plasmodium. Feeding stage. Slime molds are generally found on dry rocks, dead and decaying organic matter. Yes, they are mostly found in dead and decaying organic matter. The spores of slime molds are dispersed mainly by air current. We have discussed. They are mainly dispersed by air current. They are mainly dispersed by air currents, not by birds. So these all are wrong. So we have discussed 5 questions only, but we have ample amount of questions in that particular site. You can visit our site there. You'll get more practice question. If you want to go for online live exam, you can give live online exam. Or if you want any doubt clearing classes, you can go to this particular site. If you want to download the PDFs, mind maps, questions, worksheets, you can download from this particular map. Or if you want to directly solve your doubt, you can contact us, or you can follow our channel where you'll get updated study materials, updated questions, and updated mind maps and worksheets you'll get from this particular channel. So we have completed photosynthetic protista. We have completed slime molds, that is decomposer protista. So here in this particular class, we have completed that is called as decomposer protista. Okay, hope you all enjoyed. Thank you. Today we are going to start, that is protista, and we are going to discuss about animal protista, that is called as protozoans. We have already completed photosynthetic protistans. We have already completed that is decomposer protistans, a consumer protistan. Today we are going to learn about protozoans, which are otherwise called as the animal protozoans. So these protozoans are sometimes called as the animal protozoans. These are called as animal protozoans. So these are the protozoans. These are the unicellular animals present in protista. So the unicellular animals present in protista is called as animal protozoans. Normally they are single celled. Yes, they are normally unicellular. And they are heterotrophic nucleus, means they don't have chloroplast. As they don't have chloroplast, they are heterotrophic. Some are parasitic, means they are predator, or parasitics, means they depend on other living organism for their food. So as they are parasitic, they are normally what? Disease causing animals. These are pathogenic. Normally they are pathogenic, means disease causing, because they are parasitic in the mode of nutrition. They are believed to be the primitive relatives of animals. Yes, they are believed to is believed that they are the ancestors of animal kingdom. So normally first protozoans came into existence, and from them arises the porifera, and simultaneously the other animals arises. So for this reason these protista or these protozoans are considered as the primitive animals. And they normally contains 4 major groups. So we are going to study protozoans in 4 different groups. Let's study one by one. The first group is called as Amoeboid Protozoans, where amoeba shape-like structure is present, where pseudopodia is the main mode of locomotion. Then Flagellated Protozoans, where we find the mode of locomotion is by means of flagella. Another one is called as the Ciliated Protozoans, where we find the whole body is covered with cilia, and cilia is the mode of locomotion. And the fourth one is called as Sporozoans. Protozoans, they produce spores which are dispersed by means of air, or they produce spores that are non-locomotive, but inside the blood or inside any medium they move with the help of other vectors. So let's start one by one. So these are the certain diagram and examples. These are the Euglenas, both plants and animals we call. These are Amoeboid Protozoans. These are Giardians. We are having Plasmodium, and we are having Trypanosoma. These are in a, and we are having Paramecium. So this is in our syllabus: Paramecium, Trypanosoma, Flagellated, and we have Amoeba, and also we have Plasmodium. These 4 we are going to discuss one by one. So the first one is called as Amoeboid Protozoans. Here they are normally freshwater. Yes, they are found in freshwater, they are found in marine water, and some are found in moist soil. They are mostly free living organisms. Yes, they are mostly free living organisms, and they are parasitic. As they are parasitic, they normally are pathogenic, means they causes the disease. Pathogenic in the appearance, in the habitat. So this is the structure amoeba best example, and these are its pseudopodia. So these groups are normally the pseudopodia present that do help in locomotion. This part is the pseudopodia, and inside we find the nucleus present, the vacuoles are present, contractile vacuoles, the cell organelles are present, the food vacuoles are present, other cell organelles are present. Outside we find the ectoplasm present, within we find the endoplasm present, and within the nucleus we find the nucleolus is present, and water globules are present, ectoplasm, endoplasm both are present, cell membrane is present. But this is the basic structure of that is called as amoeba. So let's go. They capture their food, a prey food, with the help of pseudopodia, or the false food. Means suppose the food is present here, so they engulf by forming this food, and this food is called as pseudopodia. This is called as pseudopodia. So this pseudopodia, what happens? Now they engulf the food completely. They engulf and take into the body inside the cell, and with the help of food vacuoles they dissolve it and helps in nutrition. Got it? And in marine forms, in mostly marine form, silica are found on their surface. So normally the naked body means they have only cell membrane, but in the marine form some silica are found to be present on the surface. Only in marine nature this is amoeba. And the best example is Ant Amoeba histolica, which is a parasite that causes dysentery disease. We'll talk later. Next one is called as Flagellated Protozoans, and these Flagellated Protozoans are called as Trypanosoma. The best example is Trypanosoma. Normally they are free livings. They are mostly free livings, and they are found in aquatic. Got it? And they are very parasitic. They are mostly parasitic, causing disease like sleeping sickness. And these Flagellated Protozoans contains flagella as the main locomotor. So they contains flagella. So this is the flagella present. And these parasitic forms causes disease that is called a sleeping sickness. And the main causative agent is Trypanosoma. So Trypanosoma causes the disease sleeping sickness. They are flagellated because the locomotor organ is flagella, and so they are called as Flagellated Protozoans. Now let's move to Ciliated Protozoans, where we find cilia as the main mode of locomotion. So normally these are freshwater organisms, and marine forms are also present but very few in number. Very few are parasitic. Very few are parasitic normally, and they contains in the body, what happens that they contain cilia. So they contains heavy deposits of cilia, got it, throughout the body, and they contains a mouth-like opening present that is called as gullet. That is called as gullet. So by the beating of the cilia, the water enter into this gullet. The water into the gullet, and with the water, the foods are also taken in, and through this the animal engulf the food inside the body. So they contains a mouth-like structure canal that is called as gullet, and by this undulating and by this beating nature of the cilia, so the water with the food materials are moved into the gullet, and from the gullet into the body, into the cell, that do helps in its nutrition. So this is the structure of the cilia, that is the ciliated protozoans. Yes. And in this what we find, this part is the gullet part, this is the gullet part. Through this gullet part, these are the cilia present, and these cilia beats, and into the gullet the food water enters, and where the water enters, what happen? The food is taken in, and within this organism we find the food vacuoles are present, and these food vacuoles dissolves the food materials and do helps in nutrition. And like amoeba, they also contains outer peripheral ectoplasm and inner, that is called as the endoplasm. Like amoeba, they also contain so and so. So food vacuoles are present, yes. So dissolves the food vacuoles present, got it. And we find all cell organelles are present. As eukaryotic, they also contains all cell type of organelles. Okay, now let's discuss. This ciliated, yes, that one is called as the gullet, the opening outside the cell surface that do helps in engulfing the water, and through the water the food materials are engulfed with the beating of cilia through the gullet. So the movement of cilia, the rows of movement of cilia helps the water move into the body through the gullet, and through the gullet the food are also taken. So this is the main mode of its nutrition. And the best example is called as paramecium. Now let's move the last protozoans, that is called as sporozoans, and these are normally endoparasites, and these sporozoans, they include the diverse organism that they produce spore like organism, means the malaria parasite. So they produce a spindle shaped like spore, and these spores enters into the human body. Inside the body they develop into different types of spores, and then into gametes, and these gametes enter into mosquito body, and in the mosquito bodies they again form spores, and this is how the life cycle takes place. So with the help of spore they infect the human, causing the disease that is called as malaria. So within the body they causes the disease, so they are called as endoparasite, because it moves into the body within the liver and within the, that is called as the RBC, they do causes the disease. We'll discuss later in the chapter health and disease. So the most dreaded sporozoans is called as Plasmodium vivax, or this Plasmodium they call as malaria parasite, which causes the disease malaria, and that causes and that effect large human population is affected by this particular disease malaria through this organism that is called as Plasmodium. And this is the structure of the Plasmodium spores. How the spore moves into the body we'll discuss later. And this is a unicellular spore present, and within the spore present we have find microtubules are present, micronemia are present, Golgi body present, everything is present. Now let's discuss about the basic locomotory organs of protozoans. So amoeboid protozoans move with the help of pseudopodia. They move with the help of false feet, that is called as pseudopodia. And flagellated protozoans move by means of flagella. So these flagellated protozoans, they move by means of flagella. And the ciliated protozoans, they move by means of cilia throughout the body. And the locomotive structure is absent in sporozoans. So sporozoans, malaria parasite sporozoans don't have any locomotive structure. Now facts. Amoeboid protozoans contain silica-like cells in its marine form. And flagellated protozoans are rarely a sexual mode of reproduction. And these groups contains their commensal, symbiote, and parasitic in nature. Ciliated protozoans possess ingestion, means they contains the gullet, and through this gullet the food is ingested. Like animals, the food is ingested with the help of this beating nature of cilia. And the region of ingestion is called as the gullet. Yes, just we have discussed. The region of ingestion do helps in it, and that is also the mode of egestion. It is the mode of ingestion, that part, and also it has taking in and giving out. So giving out the waste material and taking in the food materials is done by this particular structure that is called as the gullet present in the paramecium surface. Infectious spores like is present in the life cycle of the sporozoans. Now amoeba causes the disease, entamoeba they causes the dysentery. Trypanosoma causes sleeping sickness in the body. Paramecium is ciliated protozoans, And Plasmodium is a sporozoans that causes the disease malaria. So now let's move into the certain practice question. We have completed the general character of the protozoans. Now we are going to discuss about some practice questions. So assertion tells that protozoans are considered primitive heterotrophic protista. Yes, they are true. They depends on the yes, the heterotrophic. Yes. So this is the right one. Got it clear? Both A and true are true and R is the correct explanations. Now consider the following statement regarding protozoans. The first statement is tell all protozoans are unique. Yes, it is right. They may be free living or parasitic. Yes. And they possess cell wall. This is not. This is wrong. And they reproduce by means of binary fission. Yes, it is found. True. So what is the correct option? The correct option is 1, 2 and 4. 1 is correct, 2 is correct and 4 is correct. Now which of the following protozoans moves with the help of pseudopoda? That is best example amoeba. The best example is amoeba. Very easy question. The locomotor organ of paramecium is cilia. The locomotor organ of paramecium is cilia. And which of the following protozoans responsible for malaria in humans? Protozoans. That is Plasmodium. So this is got it. So this is the right answer. So only we are given 5 questions here, but you'll get ample amount of questions, about 1000 plus questions about Protista. And from each section you'll get 200 to 200 type of questions, and all are of NEET level, competitive level. On exam point of view, you can go through this particular site. Here you will get maximum amount of questions, or in this particular site also you can go for online exam where you'll get the score at particular time, or you can go for only online classes for doubt clearing classes. You can go if you want to download the study materials, the mind maps, the worksheet, the MCQs, all are present in this particular site. If you want to contact, if you clear a doubt directly, you can go through this number, or you can go to these channels and clear your doubt, or you can get all the study materials, all latest questions, latest study materials you can get from our channel. Alright, so we have completed the chapter that is called as animal protista or the protozoans. So we have completed in this 4 or 5 section we have completed protista. So hope you all enjoyed. Thank you.